that, we're going to welcome Courtney Demmitt Rice. Where's Courtney? Okay. Courtney's coming to us from Josh Bunkowski's lab, where she is a neurobiology PhD candidate student. And we're really pleased that she'll be talking about, we all talk about very long chain fatty acids. She's going to talk about another lipid context to think about.
Hi, I'm Courtney. I'm very excited to be speaking to you all today. I'm really grateful for this funding and presentation opportunity. I think this is a really cool funding mechanism that this isn't a situation where money gets sent somewhere and hopefully the research ends up in a journal someday, but that the community that's giving the money, we're actually able to come and deliver this research. Link disorder, which means it primarily affects males with 3 different subtypes. So we have the adrenal subtype, the motor dysfunction subtype, which we also see presenting typically in women, as well as the inflammatory cerebral subtype. And then, of course, you can have patients that develop multiple subtypes at once, which just complicates this disease and studying this disease. So symptoms and subtypes are generally considered unpredictable, even though we know that ABCD1 mutations cause adrenoleukodystrophy and they cause accumulation of VLCFAs. There's not really a reliable genotype to subtype relationship, even with over 1,200 mutations being identified. And then the clinically available VLCFA level testing also has not given a lot of insight into what subtype a patient will develop. So just in general, how VLCFA accumulation and ABCD1 dysfunction cause symptoms is still just really not well understood. There's still a lot of work to be done. So this leads to a kind of snowball effect where when you have a disease mechanism that's not super well understood, it limits our ability as researchers to be able to develop highly accurate models, especially for disease that does have different subtypes. How can we develop models that faithfully recapitulate each subtype when we don't know how those subtypes arise? And then, of course, that leads to a further issue, which is that when we don't have great model accuracy, we have problems with developing optimal treatments. So kind of some of the current questions that I'm hoping to address in my thesis work are, what are the broader aspects of ABCD1 dysfunction that maybe haven't been explored up to date? What are some of the earliest dysfunctions that lead to disease, and can we correlate these earlier dysfunctions to developments of particular subtypes? And can we identify some better treatment options? And when I say better treatment options, I think it's so wonderful that we are really diving into gene therapy here. Hopefully we can help identify some treatments that are more accessible, less invasive, less expensive, maybe available over the counter. So I'm going to be focusing on identifying treatment options in my talk today. So to tackle this question, several years ago, our lab conducted a very large-scale drug screen of over 2,500 different compounds on our ABCD1 mutant zebrafish line, and this screen identified chloroquine as a potential treatment. We found that chloroquine treatment rescued oligodendrocyte deficiencies in the zebrafish. It also corrected aberrant swimming behavior, and it decreased VLCFAs. So this was exciting. The problem with that, though, is chloroquine is toxic. Chloroquine is a drug that we say is polypharmacologic, or maybe you've heard the term dirty drug, and that's because it has many different mechanisms of action, and it can be used to treat a wide variety of diseases. So it was originally used to treat malaria, it's been used to treat lupus, rheumatoid arthritis, and again, these very broad mechanisms of action where it's lysosomotropic, it can be anti-inflammatory, it can inhibit processes like autophagy and endocytosis. So it's really no surprise that if you were to give this drug to a patient, that it is going to cause long-term toxicity when it has so many effects. So again, it was interesting to find a drug, but it's just not something that, if you get a patient that comes back with their newborn screening and they find out that they have ALD, you cannot start giving this to a child and just have them be on it for their entire lifetime without having some serious effects. So initially, the lab had noticed that chloroquine treatment decreased saturated VLCFAs. So we looked into the enzyme that regulates that, and unfortunately, further research did not support that that was the mechanism of action by which chloroquine was working. So we're hoping that if we can uncover chloroquine's, out of all of the things that chloroquine does, if we can find the one thing that it does that is helpful, specifically in ALD, that we'll be able to provide a targeted treatment that does that same thing, and then that treatment would have fewer side effects. So again, what is the mechanism by which chloroquine rescues disease phenotypes in ALD specifically? So to answer this question, a previous postdoc in our lab, Quentin Ross, set up a multi-platform analysis. We had wild type and mutant zebrafish. They were given DMSO as a vehicle control or chloroquine, so both groups. So we had 4 groups that we were comparing. And then those groups were pooled and sent off for different types of analysis. So we did RNA sequencing on some and lipidomics on some. The lipidomics was handled by York, who you just heard from, in Stephen Kemp's lab. So they did a fantastic job on these analytics. Close to, I think, 3,000 different lipid species were analyzed. So it's really quite a treat to have that level of resolution in the analytics. So thank you to Stephen Kemp's lab for that. So when the results came back, we found that chloroquine reduced cholesterol esters. So we expected to see increases in VLCFAs in our mutant fish and then decreases. So again, that was expected. But the unexpected part was seeing these cholesterol esters increased in the mutant fish and then decreased with chloroquine treatment. So on this side of the graph, you can kind of see the salmon colored box is the wild type fish and the teal colored box is the mutant fish, and then yellow and purple are wild type treated with chloroquine and mutant treated with chloroquine respectively. So we see that there's a global increase in all or in many different cholesterol ester species in our mutant fish. And then if we break that down and look at particular species on the other 2 graphs, you can see that difference becomes more apparent. So if you look at all the different species that were analyzed, we particularly saw these differences with long chain and very long chain cholesterol esters. So I know this group is very familiar with VLCFAs, but this other lipid, cholesterol esters, what are they? So cholesterol esters are where you have a free fatty acid, so that can be short, medium, long chain, or very long chain, and it is joined to a cholesterol, and this is performed by the enzyme ACAT1. So again, it just steals a little cholesterol from the ER and joins it to a fatty acid. That's one way that cholesterol esters can occur within a cell. The other way that a cell kind of gets a hold of cholesterol esters is through endocytosis. So these cholesterol esters are circulating in the bloodstream on LDL particles. Those come into the cell through endocytosis. They get broken down in the lysosome into just the fatty acid and the cholesterol component. That stuff gets distributed throughout the cell. And then in the cytosol of the cell, we can actually have this synthesis occurring again. And once these molecules are synthesized, they can get stored in lipid droplets. And this is reversible storage, where the cell can be taking the stuff in and out as it's needed. So this synthesis and hydrolysis cycle is a typical part of healthy cellular function normally, okay? So we wanted to know, though, because cholesterol esters have been shown to be cytotoxic in some contexts, and they've also been shown to induce cellular stress, we wondered if they might be playing a pathogenic role here in ALD. So a review of the literature showed that cholesterol esters have been previously found in both pre- and post-lesional CALD tissue, also in ALD adrenal glands. A lot of these studies, though, are either from post-mortem tissue or they're from patients that are already showing symptoms. So we really don't know, just from looking in the literature, whether or not these cholesterol esters contribute to the driving a disease, or whether this is something that comes up kind of as like a tertiary symptom later in the disease process. However, with the study that we did, our fish were 7 days old, so very, very, very young fish. Zebrafish normally live to about 2 years old, so this is pretty early in the disease process. Stephen Kemp's lab also published a paper last year that showed cholesterol ester accumulation in the spinal cords of pre-symptomatic mice. So seeing cholesterol esters accumulate in our disease models, very, very early on in the disease process, indicated to us that these could be early drivers of disease. So we wanted to know, will decreasing cholesterol synthesis through ACAT1 inhibition improve disease phenotypes? So our approach to doing this is to use, again, ACAT1 inhibition pharmacologically. So we identified 2 drugs that we wanted to screen. The first is avacimbab. This drug is really interesting because it's already been approved for human use. So if it was something we found to be useful, there'd be kind of some fewer FDA hoops for that drug to jump through. The other drug I was interested in looking at is called K604. This drug has a much higher specificity for ACAT1 than avacimbab does. And it has also shown up in the literature in some other places, and it looks like it has some interesting preclinical momentum. So we decided to test this on our zebrafish model. And we did some initial dose testing, found out that doses from 1 to 10 micromolar were well tolerated in our fish. And so after finding that out, we could say, all right, so if we treat our fish with these drugs, will this phenocopy the rescue of oligodendrocyte counts? So the first dose we tested, or that I've been testing, is 10 micromolar. And I found that it did not increase oligodendrocyte counts. So if you look at the graph, the first thing you'll notice is that my mutant group, or sorry, the wild type group is gray, mutant group is pink. You'll notice you don't see an oligodendrocyte decrease in these fish, and that's one of the challenges with working with the zebrafish model, is that every time I've done it before this, it's been deficient every time. This is the first time it wasn't deficient. But I think that just perfectly recapitulates the heterogeneity of this disease. But the takeaway from the graph really, though, is that, so you have the green groups and the orange groups, those are avacimbab and K604 wild type and mutant, is that regardless of genotype, treating with both of these, or treating with either of these drugs at this concentration caused just a straight across the board decrease in oligodendrocyte counts. So in one way, this is helpful to us because we can see, this indicates to us that the drug is actually being absorbed and used by the fish. Typically, when you're treating a zebrafish model, you do a bath application of drug, which is where you just put the drug in the dishwater that they live in. So this helps us know that it is getting transdermally absorbed and is taking effects in the body. And because these results that we're seeing are not genotype specific, I think that this is a dosage issue. It's just too high of a dose, even though it was well tolerated. And so I would like to repeat this experiment with lower doses until we see some sort of genotypic specific effect being elicited. And when that happens, we can couple this evaluation or with an evaluation of myelination and behavior to assess to see if there's positive functional changes that accompany oligodendrocyte count rescue. So because the zebrafish data takes a hot minute to produce, I decided in the meantime I wanted to work in cellular models. So classical kind of ALD cellular phenotypes are going to be things like reactive oxygen species, increased ER stress, and increased mitochondrial stress. We're particularly interested starting out in mitochondrial stress. So I obtained 2 cell lines. I have some patient fibroblasts from a patient that had both AI and AMN, and then control fibroblasts that are age, sex, and race matched, and they have a normal neurological evaluation. So to evaluate this mitochondrial function, I'm using what's called a seahorse assay, a mitostress assay. So what this does is it delivers different drugs to the cells at specific intervals of time. And then it's able to measure different parts of oxidative phosphorylation that are affected by these drugs. So I set this up starting out with avacimbab. So our wild type group is in gray, mutant group is in pink. The increased color saturation in the bars correlates to increased dosage concentration. So you can see starting off that our mutant group just has way lower basal respiration than the wild type group. In the wild type group, as you see the doses increase, we do see an incremental but insignificant response to the drug. So again, that's helpful to know that the drug is getting in the cells and it's doing something. But when we look at our mutant group, we're not really seeing as much of a response to the drug, and certainly not anything that is restoring basal respiration to wild type levels. Looked at maximal respiration, saw the same thing. Insignificant incremental dose response in the wild type cells, and not much of a response in the mutant cells, and again, not rescuing to wild type levels. And also looked at spare respiratory capacity, same story here. So right now it looks like ACAT1 inhibition with avacimbab does not rescue these features. As I mentioned earlier, these results may be due to the fact that avasibimab has a much lower specificity for ACAT1 than a drug like K604. So I will be repeating this experiment with K604. And then we should also, even though these results don't look promising, it is still helpful to look at things like ER stress and reactive oxygen species, because it could be that this provides partial rescue for other aspects of the disease. So future directions. Of course, any good hypothesis has 4 backup hypotheses. So in this case, that is, thinking about the fact that ACAT1 inhibition might not be an effective treatment strategy. But again, we saw that when chloroquine rescued symptoms, we also saw these major decreases in cholesterol esters. So how else could cholesterol ester decreases be occurring? There's 3 other possible sources. So going back to the picture I showed you earlier, it could be an increase of hydrolysis in the cytosol, so that would be cholesterol ester hydrolases could be acting up more. It could be that because chloroquine is lysosomotropic, it tends to concentrate in the lysosomes, that it going there increased lysosomal function, and so there's more hydrolysis at the lysosome level. Not in this picture is also a transporter called ABCA1, and that effluxes cholesterol esters out of the cells. So it could be that this drug increased cholesterol ester efflux by ABCA1. And then, again, the 4th backup hypothesis here, which I think is really interesting, is that what if cholesterol ester synthesis shows up so early because it's actually a protective mechanism? So that's another thing that would be really interesting to explore. So with that, here's my acknowledgments. So I want to mostly acknowledge Quentin Rath, our former postdoc, who kind of got all this set up and left, and then I got to start picking up the pieces. And then, of course, Yorick Jaspers and Stefan Kemp's lab for the really amazing analysis of our lipidomics. So with that, I'll take any questions.
Thank you.
That was a really nice talk. Thank you. Going along that last question about the possibility of a protective mechanism, where do you think the substrate goes after, or if your very long chain fatty acids no longer esterified to cholesterol, where do you think those substrates would accumulate?
Oh, that's a great question. I've thought a lot about that. So there is, I'm no expert on mitochondrial function, but there is some lipid peroxidation that can occur in the mitochondria. So I don't know if there's maybe some alternate respiration of since they can't go into the peroxisome to undergo beta oxidation, or they may be going to the mitochondria and being peroxidized there instead. So yeah, that's a good question that I don't know the answer to. So,
Any other questions for Courtney? Thank you for that wonderful work and talk. Beautiful.